Hi. So I'm sorry I had to go off to New York this week, but I decided to make a streaming version of the lecture, so we'll pick up where this lecture ends off when I get back. Anyway, today's lecture is on nomenclature, names of enzymes. So we've got to name enzymes, right? Just like your parents had to name you when you were born, because if they had a second child later on in life, you had to know who they were yelling at. So Gary, do the dishes, Barbara, feed the dogs. If we didn't have names, how would we know what to do? Well, it's the same with enzymes. So if you discover a brand new enzyme, you've got to have a name for it so you can discuss it in the literature and people will know what enzyme you're talking about. So all the original enzymologists were German, so they gave German names to the enzymes. So we had names like Atmungsferment and Zwischenferment. When someone discovered a flavin enzyme, which was yellow, they called it the yellow enzyme. But then pretty soon someone discovered another flavin enzyme, so then we had to have the old yellow enzyme and the new yellow enzyme. And then there was the malic enzyme. I mean, what's that? There's no way to tell. In fact, in a lab where I worked, we just called enzymes Chip's enzymes and Gary's enzymes, and that was about it because we all knew what everyone else was working on. But you can't publish it that way. So you need to have names. So there are common names, and the common names are very slightly systematic, not entirely though. For instance, the hydrolases are usually called substrate ACE. So for instance, we have enzymes that hydrolyze amylose, they're called amylase. Enzymes that have hydrolyzed proteins are called proteinases or proteases. The enzyme that hydrolyzes lactose is called lactase. It's actually specific for beta-galactoside, so its real name is beta-galactosidase, but everyone knows it's a hydrolase. If it's not a hydrolase, you also have to name the reaction too. So for oxidoreductases, we might say substrate oxidase. So an example of that might be NADH oxidase. It could also be substrate dehydrogenase, for instance, lactate dehydrogenase. If we had isomerases, we might just name the compound that got isomerized, so triose phosphate isomerase. So all these things work. But sometimes the common names are just completely arbitrary, and there's no way to tell what the enzyme is. So some examples of this are physin, which comes from figs, papain, which comes from papaya, bromelain, which comes from a class of plants called bromeliads, like pineapple, and then trypsin, cetylysin, pepsin, chymotrypsin. All these are proteases. But how would you know what the enzyme does if you didn't already know about the enzyme? So, quite a while back, the International Union of Biochemistry commissioned an enzyme commission, and their charge was to find systematic names for the enzymes. So the Enzyme Commission is part of the International Union of Biochemistry, and what they decided to do was to classify enzymes according to the type of reaction they catalyzed, and then include more information about the substrate and the product. So what the Enzyme Commission system consists of is a number. It's always preceded by EC, so you'll know what the number means, and then four digits, I, J, K, and L. These are always integers. In addition to that, there are a set of rules for systematic names and recommendations for common names. So now what they did was to classify the enzyme according to the reaction. So that means that you might have two enzymes from different sources that catalyze exactly the same reaction, which means they would have the same name and the same enzyme commission number, even though the proteins might be completely different. No homology. One might be a monomer, the other a tetramer. They might have different active sites, but they would still have the same EC numbers. So it's like E. coli. There are dozens of E. coli. Some are benign, some are almost deadly. They're all E. coli, but they're very different. Well, it can be the same with enzymes. So we'll just classify them according to the reactions they catalyze, not to their chemical structures. So ECIJKL. I is the class of reaction. So you're going to have to be able to look at an enzymatic reaction and classify it according to the type. Luckily, there are only 6. So you'll have to know them, you'll have to be able to classify them, and then you'll have to give the value of I. So I will be an integer 1 through 6. EC1 is the oxidoreductases. EC2 is transferases, sometimes called group transferases. EC3 is the hydrolases, and EC4 are the lyases, and we'll talk about what lyases are later. EC5 includes all isomerases, and EC6 includes ligases, and we'll talk about what those are in a while. So that's I. Now J is the subclass that tries to classify these reactions, these enzymes, further. But of course the things that are important for an oxidoreductase reaction are different from the things that are important for a lyase reaction. So the values of J depend on what the value of I that you've chosen. Now in the old days, you used to have to buy a book called the something or something, first, second, third, fourth, fifth decennial index of the Enzyme Commission. And if they needed to change the names more rapidly, more often than every 10 years, they just put out a little addendum to it. But you had to buy this book and look things up. Nowadays, of course, you do it on the web, and I've given you a link on the website. But you still have to classify the reaction according to you have to classify the enzyme according to the type of reaction it catalyzes. So then you click a link that goes to EC1, and it lists all the J's and the K's and the L's. So this is still a valuable exercise, and it also helps you analyze the kind of reaction that's being catalyzed by the enzyme. So just as J depends on what I is, K depends on everything above it, and L is usually just an arbitrary serial number that the commission has assigned to that particular enzyme. Okay, so let's start with the oxidoreductases. So it should be pretty obvious that these things catalyze oxidation-reduction reactions. So their systematic names are donor, acceptor, oxidoreductase. So the donor, what is it donating? It's donating electrons. So two electrons and a hydrogen, or one electron and a hydrogen atom. The acceptor is the thing, of course, that receives that. So, in other words, the donor is the molecule that becomes oxidized, and the acceptor is the thing that becomes reduced. So there are some recommended common names for this. For instance, donor dehydrogenase. On the other hand, if oxygen is the acceptor, then donor oxidase is allowed. The subclass J identifies the chemical group on the donor that becomes oxidized. So for instance, suppose you had something that was both an alcohol and an aldehyde. How would you know whether the alcohol was being oxidized to a ketone or the aldehyde was being oxidized to an acid? They're both two electron oxidations. So that's why you need the J. For the subclasses, generally, but not always, these distinguish among the different kinds of acceptors. So cytochromes, oxygen, pyridine nucleotides, and those kinds of things. And then, of course, L is just an arbitrary but fixed serial number. EC2, the transferases, sometimes called group transferases, these take a group from one molecule, a donor molecule, and pass it on to an acceptor molecule. So the systematic names would be the donor and the acceptor, the name of the group, and transferase. The recommended common names would be something like donor group transferase if the donor was more important, or acceptor group transferase if the acceptor was more important. But sometimes, if the group that is, if the donor is ATP and the group that's being transferred is a phosphate, then you just call it acceptor kinase. So since you're saying kinase, you know that the donor is ATP and a phosphate is being transferred. So all kinases are group transferases, EC2. The subclass identifies the group that's being transferred, so it could be an acetyl transferase, a one carbon a sugar transferase, something like that. The subclasses use greater detail to distinguish among the groups that are being transferred. Is it just an acyl group or is it an aminoacyl group? Now there's one case that you might not recognize as a transferase, and these are the transamination reactions. So for transamination reactions, the enzyme, usually with something like pyridoxal phosphate, takes an amino group for an amino acid and pulls it off and leaves in its place a keto group. So it looks like the amino acid has been oxidized. But the next step is to put that same amino group back onto a different keto acid, which converts it to an amino acid. So although one is being oxidized and one is being reduced, the main thing that happens is that a nitrogen, an amino group, is transferred from one molecule to another. So these things are transferases as well. And when we talk about the coenzymes, we'll talk about that a little bit more. The hydrolases are pretty much like group transferases, except the acceptor is always water. So these catalyze hydrolytic cleavages of carbon-carbon, carbon-nitrogen, carbon-oxygen, oxygen-phosphate, oxygen-sulfur bonds, that kind of thing. The systematic names are just substrate hydrolase, as we mentioned before. The recommended common names could be just substrate ace, if it's pretty obvious what bond is being hydrolyzed. But if there's more than one bond that is labile to hydrolytic cleavage, you may have to include the kind of group that's being transferred to water. So for instance, pectin. Pectin is a polymer of galacturonic acid, so you could be hydrolyzing the galacturonic acid, the bonds between the galacturonic acid units, but it's also a methyl ester, so you could be hydrolyzing the methyl ester. So you have to specify that in the name of the enzyme. So the subclass J reflects the need to specify the bond that's being hydrolyzed. For subclasses, you need some more definition of the kind of substrate. And then a special case here is the proteases, which are peptidyl peptide hydrolases. And this is the only case where the subclass reflects the mechanism of the enzyme. There are 4 kinds of proteases, and those are the possible values of K, and we'll talk about those when we talk about proteases in a little bit. EC4, the lyases. This is probably a name that you haven't heard before. And these enzymes catalyze elimination reactions or addition reactions that are basically the reverse of the eliminations. So this is either formation or cleavage of carbon-carbon, carbon-oxygen, carbon-nitrogen bonds, etc. So the systematic names are substrate group hyphen lyase. Okay, you have to have the hyphen there. If the addition reaction is more important than the cleavage, so in other words, the enzyme basically synthesizes something, you could call it product synthase. So it synthesizes that particular product. But this can cause you some trouble because there is another kind of enzyme that is called a synthetase, which is EC6. So be very careful when you're using the terms synthase and synthetase. The subclasses contain enzymes that break different kinds of bonds. So they're going to be carbon-carbon, carbon-nitrogen, etc. And those will have different J numbers. The subclasses distinguish among enzymes on the basis of the identity of the group that's being eliminated. So not just the bond that's broken, but what's the piece that goes away. And then, of course, L is just the serial number, as it is in all of these cases. Isomerases, well, you know what isomers are. They're compounds that have the same chemical formula, but things are attached differently. So isomers have different structures. So isomerases catalyze structural rearrangements. So the systematic names correspond to the kind of isomerization that's carried out by members of the subclasses. So J, the subclass, is enzymes that catalyze different kinds of isomerizations. So racomases. So racomases invert the stereochemistry of a compound that has one optically active center. So you produce a racemic mixture. Racemic means equal concentrations of dextro and levorotatory isomers. But if the compound has more than one optically active center and the enzyme changes only one of those, they're called epimerases. They make different epimers. So like glucose and galactose are epimers at C4. All the hydroxyl groups are on the same side of the, well, have the same stereochemistry in those two sugars, except C4 where they're inverted, so they're epimerases. We also have cis-trans isomers, so there are cis-trans isomerases. We have things that differ in the position of double bonds and protons, so these are called tautomerases. They make different tautomers. And then we have mutases and cycloisomerases, things that just move groups around or change the way things are connected. The subclasses depend on the nature of the substrate, and L, of course, is the serial number. Okay, we're almost at the end. Ligases. So you probably know the word ligate from words like ligature, which is what you get when you have stitches, or maybe a tubal ligation where fallopian tubes are tied off or something like that. So this means tying things together. So this means bond formation that is coupled to the hydrolysis of some sort of high energy bond. So that means that you're forming a bond that would be thermodynamically uphill to form. So it's not likely that it's going to happen. On the other hand, if the enzyme can couple a very favorable reaction like hydrolysis of an ATP, then you can form that bond. So these are called ligases. The systematic names correspond to usually AB ligase, so that means the two things that you're putting together. But you may also have to specify how the ATP is cleaved. Some enzymes cleave off just the terminal phosphate, the gamma phosphate, and leave ADP, but others cleave the beta and the gamma phosphate off to make inorganic pyrophosphate, which means that you've left with AMP. So that might have to be specified as well. The subclasses depend on the kind of bond that's formed. For instance, carbon-carbon, carbon-oxygen, carbon-nitrogen, and so on. The subclasses are always 1. K is always equal to 1, except for carbon-nitrogen bonds, and then there is some difference there. So these are EC 6.3s. And L, of course, is the serial number. Okay, so if you look at an enzyme catalyzed reaction, you should be able to say, aha, this is a ligase reaction. Therefore, this enzyme's classification is EC6. So you have to know the number. So what my students have done in the past is come up with some mnemonic to help them remember the order of these things, and typically they come up with sentences like, well, I don't remember the sentence they come up with this, but you know, you can make up your own. So for remembering the cranial nerves, for instance, medical students have a sentence that is, on old Olympus towering tops, a fat-ass Greek viewed a hog. So the first letter of each word is the first letter of each of the 12 cranial nerves. Okay, well, we've only got 6 to do here, so make up some sentence that starts with O, and then T, and then H, and then L, and then I, and then L. Or just make up a word, Othlil. That might help you remember the order of the EC numbers. Okay, now let's distinguish before we move on the difference between kinases and ligases. So both of these involve breakdown of ATP, but kinases transfer a phosphate group to another compound. And that means that the phosphate appears in the structure of that acceptor. On the other hand, for ligases, neither AMP, ADP, or phosphate or inorganic pyrophosphate appear attached to the other products. They are just free in solution. They are products by themselves. They're part of the coupling reaction, not the bond synthesis reaction. Okay, so let's look up some examples. So I got this out of the old decyanyl index before they had anything on the web. And let's look at an example here. So EC1. So EC1 is the oxidoreductases. EC1.1 are oxidoreductases that act on CH-OH bonds, on alcohol. So EC1.1.1 uses NAD or NADP as the acceptor. EC1.1.2 uses a cytochrome as an acceptor. EC1.1.3 uses oxygen as an acceptor. And then if there's something else, it's EC1.1.99. If it's not an alcohol that's being oxidized, but an aldehyde or some other oxo group, then it would be EC1.2. And again, if a pyridine nucleotide is the acceptor, it's EC1.2.1. If it's a cytochrome that's the acceptor, it's EC1.2.2. So I think you see the way things are going here. So EC1.3 oxidizes carbon-carbon, well, CH-CH kinds of groups. And there can be a number of acceptors there. Iron-sulfur proteins, they all have different values of K. So we won't continue with this table, but I think you see how it works. So let's look at some examples here of oxidoreductase. EC1. So these are donor-acceptor oxidoreductases. So let's look at the enzyme that oxidizes ethyl alcohol to acetaldehyde. So this would be called ethanol NAD oxidoreductase. We've specified the compound being oxidized, in other words, the electron donor. We have specified the electron acceptor, and we've called the enzyme an oxidoreductase. So if you look back at the table on the previous page, I is 1 because it's an oxidoreductase. J is 1 because it's an alcohol group that's being oxidized. K is 1 because NAD is the acceptor. And L is 1 because somebody decided that the most important alcohol is ethanol. So alcohol dehydrogenase is EC1.1.1.1. So it is literally the first enzyme in the book. Suppose we have an enzyme that oxidizes formic acid, and NADP is the acceptor. So this would be formate NADP oxidoreductase. It could also be called formate dehydrogenase. And you might want to specify that it uses NADP rather than a flavin or NADH. Okay, so this would be EC1 because it's an oxidoreductase, but in this case J is 2, so EC1.2 because it's an aldehyde or a carboxyl group that's being oxidized. So formate, remember, is a carboxyl group with a hydrogen on the other side, so it's still oxidizable all the way to carbonic acid. So K is 1 because it's NADP that's the acceptor, and L is 43 arbitrarily because the donor is formate. So formate dehydrogenase is EC1.2.1.43. Okay, transferases, EC2. So again, we specify this as donor group transferase, if that's the most important reaction that we think about in metabolism, or acceptor group transferase, if that's more important. Or if it happens to be an enzyme that uses ATP to transfer a phosphoryl group, we can call it a kinase. So now we have a few limitations here. In our little reaction here, AX plus B goes to A plus BX. So clearly X is the group that's being transferred. The restriction is that X can't be a hydride, H dot dot, or hydrogen atom, H dot, because that means this would be an oxidoreductase, EC1. Another restriction is that B can't be water, because if the acceptor is water, then we have a hydrolase. If AX is ATP, then the enzyme is a kinase, but that's okay because this kinases are transferases, so it would be EC2. Okay, make sense? Okay, so let's look at acetyl-CoA acetyltransferase. So clearly the donor is acetyl-CoA and an acetate group is transferred. So this is one that I've brought up a couple of times in the class. Suppose we have an enzyme that catalyzes this reaction. We have two acetyl-CoAs, and one of those acetyl groups is transferred to the other acetyl-CoA, which makes it into acetoacetyl coenzyme A. And then, of course, we have free coenzyme A from the other one. from the other CoA, acetyl-CoA. So IC is 2 because it's a transfer. I is 2 because it's a transferase. J is 3 because it's an acyl group that's transferred. You wouldn't know that unless you looked at the book or on the web. K is 1 because it's an acyl group but not an aminoacyl group that's being transferred. And L is 9 because the acceptor is acetyl-CoA. So this is EC 2.3.1.9. How about hydrolases? Okay, substrate hydrolase or substrate group hydrolase if there's more than one hydrolyzable group. So AX, but now our B is water. So we get AX going to AH, so it gets one of the hydrogens of water, and X gets the OH. So we've used the elements of water to break this compound into two. We haven't added water to any of them. We've split a molecule in two. Okay, so 1,4-alpha-D-glucan glucohydrolase. So alpha-1,4-D-glucan sounds like starch, amylose, and it hydrolyzes the bonds between the glucose moieties, so that is a glucohydrolase. So its trivial name is alpha-amylase. And what it does is to split, by hydrolysis, alpha-1,4 linkages between glucose molecules, but it does so on the inside of a polymer. It doesn't do it from either end, but only on the inside of a polymer. So EC3 because it's a hydrolase, EC 3.2 because it acts on glycosyl compounds, sugars, EC 3.2.1 because it's an O-glycosyl bond that's split. It could be an N or something like that. And L is 1 because it produces a glucan, so other polymers, rather than either glucose or maltose. So there are three kinds of amylases, as we will see later on. One splits and just makes a glucose, so that's glucoamylase. One always makes a maltose product, so that's called beta amylase. And this is alpha amylase, which always makes two polymers. So EC3.2.1.1, and that's alpha amylase. So for the lyases, so we're going to say substrate group lyase, or product synthase, not synthetase, but synthase. So let's look at a bond to be cleaved. Well, we have elimination reactions, that's possible, or addition reactions. So these are generally going to involve double bonds. It could be carbon-carbon double bonds, but it could be carbonyls as well. It could be C double bond ends as well. So in our little addition reaction here, we have the addition of HX across a double bond. So the H goes to one side, the X goes to the other to produce a saturated compound that has now incorporated both hydrogen and the X. So if you look at the mechanism of decarboxylases, that's actually an elimination reaction. So decarboxylases, things that split off a carboxyl group as CO2, are called carboxylases. Additions to additions to double bonds or their reverse, any kind of elimination, these are catalyzed by lyases. In this case, HX can be water, but it's an addition reaction or an elimination reaction. So we're not splitting a molecule into two pieces. We're just adding the elements of water across a double bond. So in this case, HX could be water, and we would have made an alcohol, a secondary alcohol in this case. So phenylalanine carboxylase, which is a decarboxylase, would fall into this category. All the amino acid decarboxylases would. So these would be EC4 because they're lyases, EC4.1 because the decarboxylations in this case splits carbon-carbon bond, and EC4.1.1 because the group that's being split off is a carboxyl group, and EC4.1.1.53 is specific for this enzyme, which specifically decarboxylates phenylalanine. So that's the EC number for the enzyme. 5. Isomerases. Okay, so this would be substrate, and then if it's not obvious, you have to state the kind of isomerization followed by the word isomerase. So we have many kinds of isomerization reactions, as I mentioned before. We have racemases and epimerases. We have cis-trans isomerases. We have enzymes that convert aldehydes and keto bonds and turn alcohols into aldehyde groups or keto groups. So these just move carbon-oxygen double bonds around. We also have intramolecular transferases, things that might move a phosphate group from one end of a sugar to another. So these are all kinds of isomerization reactions. So let's look at ribose ketol isomerase, ribose isomerase. So it's EC5 because it's an isomerase. And what this compound does is to convert ribose, which is a ketose, into ribulose, ribose is an aldose. It converts it into ribulose, which is a ketose. So it moves the C double bond O from the first carbon to the second carbon. So it's an isomerization, but it's also an intramolecular oxidation and reduction because the aldehyde group becomes an alcohol, it's being reduced. The alcohol on C2 becomes a keto group, so it's being oxidized. Now, K is 1 because this is a specific kind of reaction, the interconversion of aldose and ketoses. And L is 20, which is specific for this enzyme because it interconverts ribose and ribulose. So EC5.3.1.20. Ligases. Okay, so you probably want some more explanation about this. This is a good time. So AB ligase or synthetase. Synthetase. So this is formation of a bond with concomitant hydrolysis of a high energy bond. So we get A plus B, and we're going to make a bond between A and B. And in order to make that reaction go to the right, we're going to have to hydrolyze an ATP. So we could do that in two ways. We could make ADP plus PI, or we could make AMP plus inorganic pyrophosphate, and it depends on how the enzyme wants to do it, or what its mechanism is. And these usually involve things that have an intermediate where the enzyme either contains a high-energy phosphate bond or maybe is adenylated. It has an AMP attached. So let's look at acetate coenzyme A ligase. So acetyl coenzyme A is a thioester, has a favorable free energy of hydrolysis. It's very hard to make. It requires about the same energy as hydrolysis of an ATP. So this is an enzyme that actually couples the hydrolysis of ATP to the formation of that thioester bond in acetyl-CoA. So it takes just acetate and ATP and a coenzyme A, and it makes acetyl-CoA. So it's acetyl-CoA synthetase. So it's EC6 because the enzyme is a ligase. It's EC6.2 because it's a carbon-sulfur bond that's being formed. And not only a carbon-sulfur bond, but the sulfur is an acid because it's a thiol, SH, which is a weak acid. And the carboxyl group is also an acid, the acetate. So this is an acid-thiol bond that's being formed. It's an anhydride between a carboxylic acid and an SH kind of acid. So K is 1 in this case, and L, which is specific for this enzyme, is 1. So EC6.2.1.1. Okay, so you got it? Okay, so I'd like to spend a few minutes talking about some specific enzymes here, and then we'll quit for this streaming lecture. We'll give some examples of enzymes from a couple of these classes. And the classes I'd like to talk about are hydrolases, oxidoreductases, and then at the end, some ligases. So I've chosen these because all the biological polymers that we think about, nucleic acids, proteins, carbohydrates, And if you think about it, even lipids, which are not really polymers, but they're composed of smaller units, all of these are synthesized by dehydration reactions, which means when we digest them, so when we're talking about nutrition or food science, they have to be hydrolyzed first. So the hydrolases are important enzymes. We want to talk about oxidoreductases because this is the most abundant class of enzymes. There are more oxidoreductases than any other kind of enzymes. And they're important because all the energy on the planet comes from basically one reaction, the photosynthetic reaction, where light strikes antenna chlorophyll, and the energy is transferred to active center chlorophyll, and an electron is promoted to a very high energy state, in other words, a reducing condition, and it reduces ferredoxin, and that gets passed on to make all the other reduced compounds in our body. So the most reduced compound in our body is fat, so that's the way we store that energy. Which means that in order to get that energy out, we have to reoxidize everything. So oxidoreductases are important. And I've included ligases just because they're spooky and it's hard for people to figure out what they are, so I'll give a few examples of those. Okay, so the hydrolases. So these are EC3 dot something. 3.1 is esterases. If these are carboxylic acid esters, regular old esterases, these include various kinds of enzymes, and there are about 70 of those, 72. If we have other esters that include phosphoesters, phosphomonoesters, like things that have been phosphorylated by ATP, we also have about 70 of those enzymes. If we have phosphodiesters, things where a phosphate links two groups together, two alcohols together, like nucleic acids, between the 3' and the 5' groups of the ribose or deoxyribose, there is a phosphodiester. So we have about 25 or so of those. Believe it or not, there are also sulfate esters. So we have sulfuric esters, and there are 16 or 17 of those. Now, if we get really specific and get down to nucleases, RNases, and DNases, these are abundant enough and important enough to have their own subclass. Sub-subclass. So they're all EC3.1 dot something. If we have EC3.2, these are glycosidases, and these include alpha amylase, beta amylase, cellulase, polygalactorinase, all of these things, lysozyme. We also have ethers occasionally and epoxides, which are like ethers except the oxygen bridges between two carbons. So these are EC3s, and then hydrolases that hydrolyze peptide bonds are EC3.4. So what might you want to know about these kinds of reactions? Well, one thing you might want to know is what kind of bond, which bond is actually split. If you want to know something about how this enzyme works and you have an ester, when you're finished you have an acid and an alcohol. And both of those things, both of those products have an oxygen in them. So which is the new one that comes from oxygen and which is the old one that was already there? So if you split at position 1, that means that the water oxygen will be attached to, will be part of the carboxylic acid, and the hydrogen will go with the alcohol. If we split at bond 2, that means that the oxygen that's existing there as part of the ester will go with the carboxylic acid, and we'll get the OH from the water, and that will be part of the alcohol. So we have two possibilities. One is the nucleophilic reaction, which is reaction 1 down here. So we make a tetrahedral intermediate and do it the normal way, protonate the leaving group with the hydrogen from water. But we also have an electrophilic possibility. And in this case, we have what looks like an elimination reaction. So how could you tell? Well, let's take a clue from a similar reaction. Suppose we had an amide. So an amide, we're not going to have that problem because in the product we're going to have an OH on one of the products, and the other is going to be an NH. And we know that when the hydrolysis occurs, the OH goes with the carboxylic acid, and the proton goes with the leaving group amine. So why is this so obvious? Well, because you can tell the difference between oxygen and water and the nitrogen that's in the amide group, that's in the amine. So we just need a way to distinguish between different kinds of oxygen. So let's use a different isotope of oxygen. So there are 3 common isotopes of oxygen, sort of common. The most common is O16, by far the most common. There's O17, which is not very common, very hard to purify, but it has a nuclear spin. That's good. But the most abundant of the 2 less abundant isotopes is O18. So none of these is radioactive. O17 has a nuclear spin, so you can tell where the O17 is. But O18 has 2 more neutrons. So O16, O18, and O17 can all be distinguished using mass spectrometry. So all we need to do is to use O18 water. We can buy O18 water, we let the enzyme work, and we essentially always find that the O18 ends up in the acid. So that means that it must have been the nucleophilic mechanism that occurred, since the O18 is part of the acid, and we don't know for sure, but the hydrogen, it looks like, the only place it could come from, the hydrogen from water goes with the alcohol. So that's strongly in favor of mechanism 1 on the previous slide. How about glycosidases? So we have a similar problem here because we're splitting a carbon-oxygen-carbon bond. So how do we know which bond is split? Well, we could do the same experiment, and essentially always we find that the oxygen 18 goes with the reducing end of the sugar on the left, which means that it attacks C1 of the sugar on the left, which we haven't really defined yet. So we have another thing that we might want to consider, and that is the specificity of these enzymes, or the efficiency of these enzymes. So if we're naming an enzyme based on its substrate, and the enzyme can actually catalyze more than one reaction, how do we know what the name of the enzyme is? Well, we've already discussed this when we talked about xylose isomerase. Clearly, if we have a low Km for a particular substrate, it's more likely that that's the real substrate if that substrate is actually around in the cell. So we have to think about sort of a ratio of the normal concentration of the substrate in the cell in relation to the Km. So if we have a lower Km for the substrate, that means binding is better. On the other hand, suppose the substrate binds and Vmax is very low for that substrate. And in fact, if Vmax were 0 for that substrate, we'd call it an inhibitor. But clearly, if the enzyme has a high Vmax for a particular substrate, that's good too. So we have to concern ourselves with both things, the strength of binding and the velocity of catalysis. So if we can measure Km or Km and either Vmax or Kcat's probably better, then we can take the ratio Vmax to Km, and we could call this either the efficiency factor or the specificity factor. Now this is also sort of a mystical thing because Vmax over Km is the first order rate constant for an enzyme catalyzed reaction at very low substrate where the reaction is first order. So that means that we could either measure Km and Vmax from Lineweaver-Burke experiments or Lineweaver-Burke plots, or we could just compare a bunch of substrates at the same concentration and measure relative rates, and as long as that concentration is less than Km, we're actually measuring Vmax over Km. So that's what we could do. Just let's compare some substrates. Let's make systematic changes in the substrate and determine what the enzyme really likes. What does it need to bind well, and what does it need in order to catalyze the reaction well? So let's look at some glycosidases. So how about specificity for a site of cleavage? So if we have this this glycoside here, which looks like maltose, and we're going to hydrolyze it by the attack of water. Let's look at maltose and compare some enzymes. So the mechanism of these typical glycosidases involve attack at the reducing carbon of the non-reducing side, and it's going to break the C1O bond, and it's not going to attack, in this case, C4 of the sugar on the right. It's going to go the other way. So amylase, cellulase, lactase, pectic hydrolases all attack carbon 1 of glucose in amylose or cellulose, or the galactose in lactase. So there is one bit of confusion here, because just regular old sugar is sucrose. And sucrose is a disaccharide, but the reducing side of glucose is part of the bond that links them together, but also the reducing side of fructose is involved in that bond that holds them together. So sucrose is both an alpha glucoside and a beta fructoside, since both sugars are donating their reducing ends to the bond. So invertase, the common enzyme that we use to hydrolyze sucrose, catalyzes the hydrolysis by attack on C2 of the fructose, which means that invertase is really an enzyme that is specific for beta fructosides, and it doesn't care so much about what the leaving group is, what the other sugar is. So if we had a mannose there instead of the glucose, since the enzyme attacks the fructose and hydrolyzes the other bond, it actually binds the fructose in the active site, and the other group is just the leaving group. There is an alpha glucosidase that catalyzes hydrolysis of sucrose by attack on C1, but that's not specific for sucrose, because it doesn't care whether it's fructose or some other sugar that's on the right. Okay? So both enzymes catalyze the same cleavage, but they break different bonds, and if we used O18 water, the O18 would appear in different products. Okay, so let's get a little more specific with our naming here. We have some nomenclature for reducing saccharides, and as I've been showing them here, the one on the left, the one that contributes its reducing carbon to the glycosidic bond, is called the glycone, and the one that doesn't, it presents some ring oxygen to the glycosidic bond, is called the aglycone. So one of them is the glycone, and the other is the aglycone. So now we can determine an enzyme specificity for the glycone or for the aglycone just by keeping one constant and varying the other. Then we could measure kcat over km. So for instance, beta-galactosidase, regular old lactase. Let's vary only the aglycone, so we'll keep galactose on the left, and we'll change the aglycone to various things. So if we start off with regular old lactose, we can measure km and Vmax, and we get a Vmax over km of 3.5. If we change the glucose into just phenol, it turns out we still get relatively good binding. In fact, binding is a little bit better. And not only that, but Vmax is a little bit better. So it looks like that the enzyme really, really likes to have galactose as the glycone, but it doesn't care so much about what the glycone is. It likes galactose as the glycone. It doesn't care so much what the aglycone is. But the paranitrophenol group is a very good leaving group, and phenol is better than glucose. So if we make better leaving groups there, the reaction is going to go a little bit faster. Even the methyl galactoside is split, but I don't really have the data. So the interpretation here is that changing the aglycone doesn't prevent the enzyme from splitting. In fact, the nature of the aglycone does have an effect on the relative specificity, but it's probably because if you make a better leaving group there, the reaction goes faster. So Vmax is high for phenolic aglycones because they're good leaving groups. Binding of the methyl acetal, this last one here, is poor, probably because the CH3 lacks the bulk of the aromatic or the sugar compounds, and methanol is might not be as good as a leaving group as the other kinds of things. So what about the glycone? it turns out if we put any sugar other than galactose there, or change the linkage from alpha to beta, or beta to alpha, we'll find that the enzyme doesn't work. So that's why lactase is really called beta-galactosidase. It doesn't care what the sugar on the right is, as long as the sugar on the left is, the glycone, is a beta-galactoside. So we could do this for other enzymes as well. For instance, we could look at maltose. So this is the enzyme that maltase is the enzyme that hydrolyzes maltose, which is glucosyl alpha-1,4 glucose. This is a structural unit of starch. Maltose is an alpha-glucosidase, and it really likes to have an alpha-glucose on the left as the glycone. Cellobiose is just the opposite. It still likes a glucose there, but it absolutely requires a beta linkage. So it is a glucosyl beta-1,4 hydrolase. It's a beta-glucosidase. How about the ring size? Well, you can put in sugars with different ring sizes, and here's some general results that come from those kinds of experiments. We're not going to dwell on this very long. You could attach the sugars together in different ways. Well, clearly all of these enzymes require that you have a bond to the glycosidic carbon, to the glycone. But for the aglycone, you can put it other places and it doesn't really matter quite so much. Now we can also talk about enzymes that have polymeric substrates. So some of these enzymes like to bind polymers in the middle, and others like to bind them on the end. So let's look at cellulase. So let's start with the smallest thing that looks like cellulose, which is just the disaccharide cellobiose. And we're going to compare this to the fastest rate we can get, which is cellopentase. If we call that relative rate 100%, then cellobiose isn't cleaved very well. The relative rate is small. Cellotriose is faster, cellotetrose is faster, but it appears to prefer cellopentose. So it has a particular size that it prefers. The same is true for pectic enzymes. So polygalacturonase actually prefers very large polymers of pectin, degree of polymerization of greater than 100, so more than 100 galacturonic acid units in this. So these enzymes probably bind more than one of the sugar units in their active site or on part of the enzyme, because they prefer the larger substrates. Now, if it were something that bound on that hydrolyzed only one sugar unit off the end, then maybe that wouldn't be so necessary. That maybe you could have a sugar that just bound the terminal one and didn't care so much about how many other sugars were attached to that. But in these natural hydrolases, that's the way it is. So we might also want to know where the enzyme splits a polymer. So we have a huge polymer of glucose. Let's say we have starch. Well, it's got a reducing end, a non-reducing end, and all of these units are attached by the same kinds of bonds. They're all alpha-1,4 glucosidic bonds. They're all potentially hydrolyzable by the same enzyme. So does that mean every bond in this polymeric substrate is hydrolyzed at the same amount, at the same rate? And the answer is no. So most exoglycosidases, which start from one end, usually the non-reducing end, almost all that I can think of start from the non-reducing end. But endoglycosidases cleave only interior glycosidic bonds, and they have very low rates for small fragments, because small fragments don't really have ends. Right? If you have a large polymer that has an end, it's clear what the end is. If you have a large polymer, it has a lot of middle places for the enzyme to cleave. So we'll talk about amylases, but I think my time is up and I have students coming, so I'm going to stop right here. So you guys have a good 3-day weekend, and I'll see you when I get back. Bye.